The overall goal of the following experiment is to analyze the structure and function of pure ribosomes containing a unique peptidyl-tRNA. This is achieved by performing in vitro translation assays with release factor-depleted cell-free extracts and biotin-labeled mRNAs to generate stalled peptidyl-tRNA ribosome complexes attached to the biotin-labeled mRNAs. As a second step, streptavidin-bound paramagnetic beads are mixed with the in vitro translation assays to allow their interaction with the biotin molecules located in the mRNA. Later, the beads are extracted using magnetic fields, which pull the stalled peptidyl-tRNA ribosome complexes attached to the mRNAs out of the total reaction. Next, the purified peptidyl-tRNA ribosome complexes can be mixed with translational factors, such as release factor, translation-inhibiting antibiotics, or chemical modifying agents in order to analyze the function, inhibition, and structure of the ribosome. Results are obtained that show inhibition of hydrolysis of the peptidyl-tRNAs and its correlation with structural changes in the ribosomes, based on peptidyl transferase activity or translational termination assays using SDS or tris-trypsine page gels, and modification protection assays using urea page gels.
The main advantage of this technique over existing methods like differential centrifugation is that the isolated samples mostly contain ribosomes with only one population of peptidyl-tRNAs.
This method can help to answer the key questions in the gene expression field, such as the nature of nascent polypeptides and antibiotics affect the ribosome function, like modulating the translation process.
To begin this procedure, obtain 2 grams of a dry E. coli bacterial pellet from mid-log phase cultures. Wash the pellet by resuspending in 1 liter of butter.